Good afternoon. Okay, so what we're going to cover today, and the big question that I want to ask is COVID a living thing? And it may be a touchy situation because obviously we all have been impacted by the virus, COVID. And for this lesson, what we're going to do is just compare viruses against bacteria or other cell types. And so moving right along, is it a living thing? And so we know that prokaryotes, also known as bacteria, are different from eukaryotes, which are also known as animals or plant cells. We know that they're different in some of the structures that they have and some of the things that prokaryotes lack. And we especially know that prokaryotes and eukaryotes are both considered living things. However, what about viruses? If you look at the images, right, you see, the cells that I have out there, the animal cell and also the bacteria cell. But look at the virus. Does it look like it's a living thing? It actually looks more technological than living, okay? So with the virus, some of the similarities that it has to living things that argue for it possibly could be living is the fact that viruses have DNA and RNA inside of its shell. If you look at the very top, that little circular hexagonal shape, inside of that host DNA and RNA. It also can evolve like other living organisms. It also is responsive to its environment. And so since it responds to its environment, one could argue that it could be living. And then it also has glycoproteins. And glycoproteins, we will cover a little bit more in depth in class, but for the simple definition, glycoproteins are those that sit on the cell membrane of animal, eukaryotic, and prokaryotic cells. And since they sit on the membrane, they act as receptors. And so glycoproteins for viruses are, in a sense, the same thing, receptors in order to get into the host cell, right? And so moving right along, viruses are not considered living things, though. Even though they have a lot of similarities to living things, such as the DNA, the glycoproteins, it being able to evolve, they're not, because they cannot reproduce without a host cell. And so the way in which viruses insert themselves into a regular body cell, then what it does is it releases its information to then start replicating more viruses. And then once it replicates enough, the cell will then burst open, and now you have a bunch of different viruses that can then go inhabit other cells and reproduce that way. It cannot reproduce without its cell, and that is a very key factor. It cannot reproduce without a host cell, okay? Another difference is it does not have organelle. You know how we were saying in the endosymbiotic theory that mitochondria and chloroplasts came from a prokaryote way back in the day? Well, those are organelles, mitochondria and the chloroplasts. And so those organelles are what help the cells in eukaryotic cells produce energy and survive, whereas viruses do not have any organelles. And then lastly, the last difference that is between a living thing and viruses is viruses can survive what is called crystallization. And so when we use the word crystallization, I want you to think of frozen, okay? So if you remember, or if anybody's watched the Captain America movies and how he was frozen in ice and then he was thawed out, okay? That frozen, right, it being frozen solid, is very similar to the crystallization process of what viruses do. Living organisms, we cannot survive that crystallization process. So that is another difference, okay? And so the last thing that I would like to go over is the anatomy of a virus. And so we're going to mainly focus on the influenza virus, which is the circular looking virus, okay? And so as I said, that similar to living things, they have their own DNA or RNA. And in the image, you can see what is seen as RNA, right? You can see it being labeled RNA in the center of the picture. However, you also can see that there is a viral envelope. And this is also very similar to living things because we have a nucleus envelope in our cells, which is an envelope that separates the nucleus from the rest of the cell. And so the viral envelope, in a sense, does the same thing. It protects the DNA or the RNA in the center of the virus and only opens when it needs to and only lets the RNA or DNA out. And then the last two things are the protein spikes. If you guys have listened to any of the COVID news, we always are talking about the spike proteins. The spike proteins are what allow the virus to get entry into a cell. And so these spike proteins, also known as glycoproteins, are present on both eukaryotic and prokaryotic and viruses. And that's what allowed them to get in. And then the last and possibly the most important structure of a virus is the protein caspid. The protein caspid is very similar to the viral envelope in which it protects the RNA. So if you think about it as an onion, right, there are many different layers. The caspid is the very central layer, the last wall of defense in compared to the RNA that holds the RNA, okay? And so the protein caspid is very unique to viruses. Only viruses have protein caspids. And so if there were a question to ask you about the different structures of a virus would be identified by its protein caspid, okay? And so just to reapproach the question, we are asking whether or not COVID is alive, right? It's not, because it's considered a virus. So therefore, antibiotics and other medical treatments will not help it. The only thing that we can do to combat viruses is create vaccines. And vaccines are what allow us to become immune to the virus and build an immunity to it so that we're able to combat it and we are able to stop it before it inhabits host cells and it replicates. So as always, we are going to go over this a little bit more in depth in class. Rewatch this video as many times as possible, and also bring your questions to class. That way we can talk as a class and we can kind of go over some of the details of the viruses and their characteristics.